the same adult organism. Because of the advantages and disadvantages between asexual and sexual reproduction, some organisms are able to choose. These organisms can switch between sexual reproduction and asexual. And some of them have periods where the adult organism that's produced through asexual reproduction looks different from the organism that's produced during sexual reproduction. This is referred to as alternation of generation. An example of these type of organisms would be ferns and mosses. They have both a haploid stage, which is when only half of the chromosomes are present, commonly referred to as the sex cells, as well as a diploid stage, which is when 2 sets of chromosomes are present for every trait. Each of these stages has an organism that's produced. The picture that you see below is a diploid stage of a fern plant. In alternation of generations, this fern plant, this diploid, will produce spores, which are haploid cells, or sex cells. Those spores will then be carried to a different area. And the spores will grow into small haploid organisms, which look like very, very, very small plants. Those haploid organisms can then produce haploid male sex cells and haploid female sex cells. When the haploid male and female sex cells combine, we have the diploid, which is the typical fern that we're used to seeing. The fern plant will continually go through a diploid stage, where we see the large fern plant that has twice the number of chromosomes, produce spores, and grow into haploid plants that produce cells that combine to once again form the diploid fern. In most of the cases of alternation of generations, the diploid stage is normally what we call dominant, which means these are the larger plants, the larger organisms. In comparison, the haploid organisms are normally very small. This means that the fern plants are able to take advantage of all sexual reproduction has to offer, as well as asexual reproduction. They can reproduce very quickly through asexual reproduction, forming many identical organisms, but they can also increase their genetic variation by going through sexual reproduction. Even though single-celled organisms like prokaryotes create identical copies of each other, they also want to be able to increase their genetic variation. So they've created a separate process of exchanging genes. This is referred to as conjugation. And sometimes it's thought of as a form of sexual reproduction in microorganisms, but it does not produce any new cells. In conjugation, one microorganism, one bacterium, would grab onto another bacterium using its pili, which is a small tube that extends from the bacterium. The pili grabs the other cell, brings it near, and forms a bridge. This connection allows for small segments of the DNA to be exchanged. Plasmids are a small ring of DNA that are present in some bacteria cells. Plasmids would contain an extra set of genes that give an advantage to the bacterium. For example, some bacteria contain plasmids that have antibiotic resistance, so that when they're exposed to antibiotics, they're not affected by them. This would be an advantage for the bacteria cell. The plasmid can copy itself, and when the pili connect the two cells, the copy of the plasmid goes through the pili into the other cell. The pili will then disattach from the other cell, and the two cells can go on their own separate ways. So even though no new cells were created, conjugation increases the genetic variation among bacteria because they're able to exchange segments of DNA.
I don't think it really